Bismillah, Assalamualaikum warahmatullah. You're listening to microbiology lesson on microbial adaptation. This is part 2. In part 1, we covered signal transduction and enzyme regulation through induction and repression. Now we're going to cover two more modes of adaptations, which are quorum sensing and taxis. Let's talk about quorum sensing first. So, one of the most fascinating things I've learned about microbiology is, especially the first time I've read it, is the fact that microorganisms have a social life. That's the kind of things that before you learn it, it didn't occur to you. And once you have learned it, you'll be like, of course, how could it be otherwise? Right? So that's why I like learning stuff. Yeah. So just like us, microorganisms can't survive without friends. It's part of their survival adaptation. It's an adaptation strategy based on population density. As a population, they need a quorum before they produce certain toxins, enzymes, adhesive molecules. The word quorum means, for example, in certain organizations like a law firm, before they adopt a key decision, they need a certain number of senior partners in the meeting. That number is a quorum. If they don't get that number, if they can't assemble a quorum, the firm can't adopt that new decision, even if everyone present agrees with the decision. So in microbiology, the name quorum sensing comes from that. You need to hit a certain number of microorganisms before we can observe certain behaviors in the population. And the most common manifestations of quorum sensing is the formation of biofilms. They are everywhere, in dental plaques, in slimy layers that you can see on rocks. Doctors have observed them even inside our lungs and other organs in cases of cystic fibrosis, which is the biofilm of Pseudomonas aeruginosa. So the formation of biofilm begins with a couple of bacteria, free-floating bacteria, decide to settle down to land on a surface. After they land, they release biochemicals that help them attach themselves to the surface so that they won't be swept away by fluids in their surroundings. It's their version of building a home on a land they just found. By the way, if you watch loads of sci-fi shows or you play a lot of sci-fi games, when you study mechanisms like this, often your mind starts to fantasize about, for example, alien stories, how this is like an alien species landing on a planet and construct a fluidic base for themselves, you know? When that happens, when your mind triggers some kind of lateral creative imageries, you might have the urge to push it away, especially if you have a serious conservative personality. You feel like, I don't want to be distracted by that rubbish. I'm studying right now. I'm a serious student. My advice is, don't do that. Don't push that away. Learn to embrace that imagination. You don't have to tell people about it, about your crazy, silly imaginations that you use to study something. You just use it for yourself. Because those imageries can help your memory, it can help your comprehension, especially when you do your PhD later, or even before then, I think, when scientific concepts you're learning get abstract and complicated. You can use those imageries that your mind conjures up to help you remember and understand the scientific concepts. Okay? Anyway, let's go back to this. So the bacteria land on the surface, releasing adhesive matrix that helps them stick on the surface. And they start releasing signaling molecules called inducers. Some of these inducers may encourage more and more bacteria to join them to make the biofilm thicker and larger. And when they get a quorum, when they get a certain number of cells, those inducer signaling molecules trigger the expression of certain genes. At that point, they start to behave as a collective. They secrete their enzymes in unison or produce biomolecules that they wouldn't have produced before they reach the quorum. Once they reach a quorum, biofilm starts to display properties and behaviors that we didn't see before. I want you to remember three of them. Number one, biofilms start to be resistant to antibiotics. I'm talking about the exact same antibiotics that would have killed the individual members of that community. One way they do that is by forming colony wrinkles. When cells at the bottom of the colony die, the dead cells buckle upwards, like fold upwards, so the colony surface is not flat anymore. They have ridges. It's like when you put a carpet on the floor of a large masjid. The carpet gets that kind of ridges and bulges, and you need another guy on the other side of the masjid to help pull the carpet to flatten it. So it's like that. The colony has wrinkles because of the buckling of dead cells underneath. When this happens, the area becomes thick and hard for antibiotics to penetrate. So the colony becomes antibiotic resistant. The second thing that happens to biofilms is you've got these lateral exchanges of genes. So even when you have a biofilm consist of different species, you're going to observe them trading genetic materials with one another. And that's a remarkable social behavior because it's not only among different strains, you see. It's not like us trading survival materials between races, like the Chinese trading with the Arabs and the Arabs trading with the Spanish. It's more like us human beings actively trading with eagles and the eagles trading with oak trees, like different species. So that's how remarkable the level of communication is within biofilms. So the bacteria execute that genetic transfer through transformation, transduction, or conjugation. Those common three mechanisms you've learned already in microbial genetics, which are through taking in exogenous nucleic acids, that's transformation. Transduction is transfer through viral vectors, which is different from signal transduction, like I mentioned in the last episode. And conjugation, which is through direct cell-to-cell contact. The third thing that occurs in biofilms that you wouldn't see in individual cells is sibling warfare. Sibling warfare is a bit like civil war that we observe in humans. When we split a microbial colony into two, and they each start their own biofilm community, what you'll see is they start to behave like rival factions. They will detect each other's smell, the gaseous release from each other, and then they will grow away from each other. Even if they are siblings, even if they come from the same parent colony. The communication between cells in a quorum community involves autoinducers. And one of the reasons why the number of cells matters in a quorum is because the number of adjacent cells will determine the concentration of autoinducers. Autoinducers can use the two-component regulatory system that we learned in the last episode. A good example of this is AHL, or acyl homoserine lactones. AHL plays a role in bioluminescence in Alivibrio fischeri. It helps the bacteria to glow. That glow is generated by luciferase enzyme. And that enzyme is produced following the activation of an operon called lux operon. In lux operon, beside the structural genes which make the luciferase itself, lux operon has lux R and lux I. Lux R makes the activator protein. Lux I makes the AHL synthase. The role of AHL synthase is to make AHLs, the acyl homoserine lactones. When the cells achieve a quorum, AHLs will induce the activator protein. And that activator protein will bind to a regulatory site called lux box. And that binding activates the whole lux operon to produce luciferase. Okay, so let's go over it one more time. Lux operon has lux R and lux I. Lux R makes the activator protein. Lux I makes AHL synthase. When the cell number is too low, the Lux or activator protein is not active. AHL synthase, on the other hand, it keeps making AHLs. Some AHLs stay in the cells, and some others are released to the adjacent cells. When you have more cells, when you reach a quorum, you have more AHLs. At a certain level, AHLs are concentrated enough to induce the Lux or activator protein. When this happens, the activator protein will bind to Lux box. Lux box is a region on a DNA. This binding activates the whole operon, which means it starts the transcription of the structural genes called Lux CDA-BEG. That transcription helps to produce luciferase, and that luciferase makes the bacteria grow. So that's the mechanism of Lux operon. The last thing we're going to touch regarding microbial adaptation is taxis. Taxis is microbial movements in response to something, in response to environmental stimuli. The primary one that you should know is chemotaxis, which is the movement of microorganisms in response to chemical signals. Now, note that we also use that word chemotaxis in immunology as well. When we talk about our leukocytes moving towards the site of infection, they move there because of interleukins and other chemical signals. So here, chemotaxis happens when microorganisms move either towards attractants or away from repellents. Beside chemotaxis, there are other forms of taxis, some of which you might have not heard before. The first one is phototaxis, which is instead of moving in response to chemical gradients, the microorganisms respond to light gradients. They move towards an area that's brighter. The second form is scotophobotaxis. Now, the difference between scotophobotaxis and phototaxis is subtle. In fact, some scholars just group them together. They call scotophobotaxis a kind of negative phototaxis, but I don't see it that way. So here's the difference. With phototaxis proper, the microorganisms move towards the higher intensity of light. It's directional, almost intentional, based on light gradient. With scotophobotaxis, what we observe is the microorganisms don't seem to move directionally towards the higher intensity of light, but when they happen to swim outside of illuminated area, when they happen to swim into darkness, they tumble. They swim back in a run away from the darkness. There's actually a psychological condition called scotophobia, which is the extreme fear of darkness. So people with scotophobia, if there is a blackout, they become incredibly anxious. Their heart rate goes up. I imagine that's where microbiologists got the name from for this form of taxis. So again, for phototaxis, it's like microorganisms are saying, wow, it's brighter there than here. I'm going to go there because I like lights. So that's phototaxis. For scotophobotaxis, they're going to say, yeah, it's brighter there, but it's bright here too. So it doesn't matter. But when they accidentally swim into dark areas, they'll be like, oh God, it's dark here. What's going on? I need to swim back. Right? So that's scotophobotaxis. That's the difference. Other forms of taxis are aerotaxis, which is in response to gradients of oxygen, hydrotaxis, where some cyanobacteria move towards water. You have osmotaxis, which is movements in response to osmolality, and osmolarity as well, I suppose, and many others that you can explore yourself later. The mechanism of adaptation through movement is well studied in E. coli. So scientists look at their chemotaxis in particular, and then they can reasonably induce similar mechanisms in other forms of taxis as well. This movement involves flagellar motility. A flagellum is a protrusion. It's like a thread on the surface of bacteria. Some eukaryotes have it as well, but the prokaryote ones are smaller, around just 10% the size of eukaryotic flagella. From the outside, a flagellum is made up of a filament, which is attached to a hook. The hook is attached to a shaft, which is embedded within the cell wall. The shaft is then connected directly with an inner protein ring, which is located at the edge of cytoplasm. So a few of these flagella can bundle together, and the inner protein ring that I mentioned is part of flagella motors located on the plasma membrane. These motors can spin either clockwise or counterclockwise. And the motors affect the rotational force and direction of the filaments outside of the cell, and that determines the flagella motility. If the rotation is clockwise, they tumble. If it's counterclockwise, they run. So when the flagella don't bundle together, each flagellum spins independently. When this happens, the microorganisms tumbles about, kind of move around randomly. And at other times, the flagella would bundle together and spin counterclockwise and push the bacteria towards one direction. We call this movement a run. And they can run pretty fast too, up to 20 times the length of their body in 1 second. And whether they do one or the other, it depends on the gradients of the particular attractant or repellent. So if there is no gradient, the microorganisms will just tumble and run in no particular direction. But say you have high concentration of attractant on the left side, you're going to observe the microorganisms using the tumbling movement to, orientate themselves towards the left and then run towards the left. That's what we call positive chemotaxis. The negative chemotaxis is the opposite. If you got repellent on the left instead of attractant, the microorganisms will tumble around a bit, trying to orientate themselves towards the right direction. And then they'll swim there. They'll run to the right, away from the higher gradient of repellent. Okay, we're going to end here. I'm going to give you a bit of homework. I rarely do this because I know homework can be a repellent for you. You're going to tumble and run away. But I'll give you some notes to accompany this paper that I want you to read. It will help. But at the same time, I'm not hand-holding you. I'm letting you grow and strengthen your brain cell connections. It's on how rotational direction works for the flagella motors, based on phosphorylation and dephosphorylation, a bit like the signal transduction mechanism that I taught you in the last episode. Alright, that's it for this topic. Keep on reading, keep on learning. Talk to you next time. Barakallahu li wa lakum. Wassalamu alaikum wa rahmatullah.